scientists may be a step closer to finally explaining why some allergies can last a lifetime while others fade. Turns out, the persistence of allergies may be linked to a unique type of immune cell. Allergies have long puzzled scientists. Researchers don't fully understand why allergies affect some people but not others, or why allergies emerge in the first place. Previous research has found that the type of antibody most often associated with allergies is produced by cells that don't last very long in the body, which makes people's lifelong allergies harder to explain. 2 new studies, published back-to-back in the journal Science Translational Medicine, might help solve that mystery. The studies, one of which examined children with allergies and the other adults, described a unique type of immune cell that hasn't been tied to allergies before. The cells typically produce a type of antibody not associated with allergies, called immunoglobulin G, IgG. But a subset of these cells actually switch over to making an allergy-related antibody called immunoglobulin E, IgA, when confronted with an allergen, whether pollen, pet dander, or peanuts. IgA is usually produced by short-lived plasma cells, which churn out antibodies as an immediate and temporary line of defense for the body. These antibodies are thought to help fight off parasites, for instance, but in allergies, they go after harmless proteins instead. The newly described cells are a type of memory B cell, which typically remember viruses and bacteria and spit out IgG when those invaders show up. But now, scientists have uncovered a subset of memory B cells that remember allergens and can make IgA. These cells are not short-lived like plasma cells and instead linger in the body for an indefinite amount of time, many years, or possibly a person's entire life. The new research could be helpful in developing new treatments or tests for allergies, for instance, to assess if a childhood allergy is likely to persist into adulthood, the study researchers say. These cells may be analyzed as sort of a biological marker for risk of allergy or allergy persistence, said Maria Curato de la Fale, a professor of pediatrics and of immunology and allergy at the Icahn School of Medicine at Mount Sinai Hospital in New York and senior author of the study on children. That study focused on children with peanut allergies and analyzed blood samples from 58 kids who are allergic to peanuts and 13 who aren't. The second study analyzed a smaller number of blood samples from adults with a variety of allergies, including 6 with a birch pollen allergy, 4 with a dust mite allergy, and 11 with peanut allergies. While all the children with allergies avoided eating peanuts, participants in the adult study with birch pollen allergies gave blood samples before and after starting an immunotherapy treatment for their allergies. This treatment is designed to desensitize the immune system by exposing patients to a small amount of allergen and then gradually increasing the dose over time. Both studies looked for signs that memory B cells might switch over to producing IgA after being exposed to allergens. For instance, researchers examining samples from children searched for B memory cells with a specific type of receptor, or part of the antibody that enables it to bind to specific proteins. This receptor is more common in people with eczema and asthma, 2 inflammatory conditions that often coincide with allergies, than in people without the conditions. The 2 studies pinpointed the same type of memory B cell in people with allergies, although notably, earlier research had found similar cells in animals and in people with asthma and eczema. The cells directly create IgA antibodies, the type that make us allergic. Joshua Koenig, an assistant professor of medicine at McMaster University in Canada and the first author of the paper focused on adults, told Live Science in an email. They're really the long-term memory reservoir of allergy. The research is part of an important area of study, and the key to understanding the persistence of diseases that are caused by antibodies, like allergies. Dr. Sarita Patil, an assistant professor of allergy and immunology at Harvard Medical School who was not involved in the studies, told Live Science in an email. Both studies were limited by having a small number of participants. Future research could look at how immunotherapy for peanut allergies impacts the newly described cells and the antibodies that they release. It's known that the immunotherapy reduces people's allergen-specific IgA levels over time, but the impact on these memory B cells is unclear. Other future research could see whether the behavior of these cells changes over time, especially in children, who sometimes outgrow their allergies. Understanding why allergies persist could one day help scientists eliminate or modify these allergy-specific cells so that they don't produce IgE and trigger an immune response anymore, Lafayette said. In other words, studies building on this research might one day help lessen the impact of allergies or even cure them. This article is for informational purposes only and is not meant to offer medical advice.